Hello everyone and good morning and welcome to today's webinar. My name is Ralph and I'm the CEO at dynamicBIOSENSORS. Today I'm here with my colleague Daisy. Daisy is Market Development Manager in our company and a proFIRE expert. And she will show you some recent development from our labs in the area of site-selective labeling of proteins with oligonucleotides and the labeling of antibodies with DNA. The title, as you see, the title of the presentation is Practical Insights into the Preparation of Pure Antibody DNA Conjugates with proFIRE. I hope you will enjoy today's presentation and get some useful insights for your labeling approaches. As an outline for today's presentation, we will start with an introduction into today's topic, including a short video about the proFIRE. And you will see hands-on how easy it works and what the features of this product are. And then Daisy will present our recently developed kits for the conjugation of oligonucleotides to antibodies and a new strategy to prepare protein DNA conjugates in a more site-selective way. And she will present some nice data from our customers where the proFIRE technology has been used in their experiments. At the end of our presentation, we are happy to answer your questions. So if you have any questions, please feel free to send the questions during the presentation in the questions section, whenever you have one. When you look into the literature, you see a tremendous number of publications coming out with the focus on site-specific and site-selective labeling of proteins with oligonucleotides. Some attempts involve complicated and harsh chemistries, and some reactions can only be conducted if the protein is modified during the production. For example, if unnatural amino acids are being incorporated. We at dynamicBIOSENSORS have put our efforts to make your lives easier by creating a kit where you achieve high selectivity and you can use your native proteins without modifying them. But not only pure protein DNA conjugates are necessary for different applications. Having well-defined antibody DNA conjugates is crucial for an increasing number of novel techniques. These techniques employ antibody DNA conjugates as a measure to increase sensitivity or capability and to guarantee good yields. And producing pure antibody DNA conjugates can increase the sensitivity of protein detection methods and broaden imaging capabilities. Here we have put together an overview for different applications where pure antibody oligonucleotides are essential in order to get good results. On the upper left, we see the schematic view of a switch-sense biosensor, our other technology for molecular interaction analysis, and to determine kinetic rate constants and many more parameters like conformational changes. Of course, we want to have pure antibody DNA conjugates for our technology. Besides us, there are many more techniques which require pure antibody conjugates. If you look on the upper right, one of the most well-known ones are proximity extension assays and proximity ligation assays for looking at protein-protein interactions. But other techniques require pure conjugates too. If you're doing site-sec, for example, or using immuno-PCR, you will need good conjugates. And also for DNA paint experiments, it's necessary to have really good and pure antibody DNA conjugates. There has been a bottleneck for the production of pure antibody DNA conjugates, and therefore we created a simple workflow for the preparation and purification of those conjugates. And we'll have a look at the workflow on the next slide. So we have developed different coupling kits using different chemistries for the purpose of doing a conjugation of a DNA to a protein or an antibody. You just have to think of your oligo. You can use any sequence between 10 and 150 bases and order it from your manufacturer of choice, and then you just need your protein. The rest for the preparation of your conjugate is included in the kit. Just follow the protocol and inject the sample into the proFIRE. As you will see in the video in a second, you just have to enter the length of your DNA oligo and start your purification run. The proFIRE takes care of the run. You can have a coffee And after 30 minutes, just analyze your run automatically and proceed with your experiments. And now we will have a look how it works in reality.
So now that you had an overview about the system, let's talk about some examples of conjugation of antibodies to DNA. In the next slides, we want to show you how you can easily and effectively conjugate protein to DNA and purify these products using the proFIRE technology. So here we would like to introduce you a conjugation kit that can effectively produce antibody-DNA conjugates using DBCO chemistry. This is an overview of the protocol, which consists of 3 steps. The first step is the DNA modification, in which the oligonucleotide is incubated with a cross-linker for 20 minutes. In the second step, the antibody of interest is incubated with the modified DNA for at least 1 hour at room temperature. The third step consists on the purification, in which the mixture is injected onto the proFIRE, and after 30 minutes, the corresponding fraction of the conjugate are taken and concentrated to the desired amount. After showing you the standard protocol, we would like to present you the molar ratio of cross-linker oligonucleotide and antibody, so how this ratio is crucial for the success of the conjugation. We're looking at both chromatograms. It is visible that the different ratios of cross-linker oligonucleotide and antibody affected the yield of the conjugation. On the first chromatogram, you can see a tall and well-defined peak on the box A, whereas on the second chromatogram, you see a much broader peak followed by a shoulder on box C. When analyzing the eluted peaks on a STS page gel, we notice that all peaks are pure, but the first protocol indicated by band A on the gel showed the highest yield. So the optimized ratios of cross-linker and oligonucleotide can be found in our coupling kit for antibodies. And we all know that the yield of the reaction is also important. The final product needs to be pure in order to avoid false positives in your assay. We have compared our conjugation and purification protocol to other conjugation kit available on the market. Conjugation and purification protocol was performed following the manufacturer's orientation. On the right side, the presence of a tall peak after fraction 13 indicates the presence of free DNA after the purification using the competitor's protocol. On the left side, when using proFIRE reagents and instrument, we can see a clear peak for the conjugate with no contamination of remaining oligonucleotides or free protein. Now I wanted to highlight a paper recently published at Scientific Reports out of the Helmut Centrum. The research group of Matthias Meyer combined non-site directed antibody conjugation technique with the proFIRE purification system to obtain single and double oligonucleotide conjugated antibodies. They optimized the conjugation reaction by evaluating the amount of cross-linker, reaction temperature, reaction duration, oligonucleotide length, and presence of secondary structure in the oligonucleotide. They were able to achieve conjugation yields of 30% at a starting quantity as low as tens of nanograms of antibody. After optimizing the conjugation protocol, the researchers performed a proximity ligation assay targeting the receptor IGFR-L1. They used purified single and double DNA conjugated antibodies and compared to unpurified conjugates. So when using unpurified conjugate, it is possible to find positive PLA dot counts in knockout cells as shown in the figure by the white arrows. Overall, the authors managed to show the importance of performing the PLA with purified conjugates to avoid false positive results. So this is also a very interesting paper that we recommend to everyone either working with PLA or developing customized conjugation protocols. In the second case study I wanted to show you is of a research group from the Karolinska Institute. They have developed a conjugation approach which combines a nanobody DNA enzyme chimera and conjugated to a G quadruplex structure. The authors describe yields of about 55% at a 1-to-1 molar ratio of protein and DNA. They have used the proFIRE to purify their conjugates. The purification could also be done by gel separation, but this has proven to be time-consuming and decrease the yield of the conjugation. The presence of conjugates as well as its activity are shown on the gel. Overall, the researchers describe that the conjugates produced can be used as a highly sensitive and convenient immunoreagent for a specific detection of the toxic alga Alexandrominutum in a peroxidase-like activity-dependent ELISA test, which can be used to monitor water quality. So we know there is also a constant need for site-selective conjugation protocols. In the next slides, we want to introduce you to a general approach to obtain site-selective protein DNA conjugates using his-tag proteins. This approach comes as an exclusive proFIRE amine coupling kit for his-tag proteins. For the his-mediated coupling, the first step is the DNA and ligand modification. In 2 separate reaction tubes, the guiding DNA is incubated with the ligand for 15 minutes and DNA nanoliver is incubated with a cross-linker for 5 minutes. In the second step, activated nanolivers are incubated with the guiding DNA his-tag protein mixture for at least 1 hour at room temperature. The third step consists on displacement of the guiding DNA by incubating the previous solution with a displacing DNA and EDTA for 5 minutes. After the purification, the conjugates are ready to use. So here we want to guide you through a chromatogram obtained after the conjugation using the kit mentioned beforehand. We conjugated 50 micrograms of GFP nanobody to a 48-mer DNA, successfully yielding 5 micrograms of conjugate. On the left side of the screen, you can see the chromatogram, which shows a well-defined peak for the conjugate and how it's separated from the free DNA. On the right side, there is a screenshot of our data viewer software that guides you in identifying the conjugate peaks and automatically estimating the amount of conjugate in each fraction when you select it. So we know that especially when it comes to sizing and detection of conformational chains, it is very important to have control over the conjugation protocol to ensure optimal results. Here, we used protein DNA conjugates obtained with the his-mediated coupling to functionalize the biosensor surface and determine the size of proteins. The size obtained for monomeric ubiquitin, human carbonic anhydrase, and protein A were 3.6 nanometer, 5.6 nanometer, and 5.95 nanometer, respectively. The same approach could also be used to determine real-time kinetics using our novel helix instruments. So as a summary of today's presentation, we demonstrated how the proFIRE system can be used to efficiently obtain well-defined protein DNA conjugates and its importance for the success of your experiments. During the presentation, we highlighted the importance of having pure antibody DNA conjugates to increase sensitivity. We have also presented a his-mediated coupling kit for site-selective conjugation. So we want to stress that the proFIRE technology goes beyond antibody DNA conjugates. We also want to encourage you to visit our website to know more about other conjugation tools and applications of the proFIRE technology. There is a lot of information, not only about the proFIRE, but also about the switch sense there. So thank you very much for being with us today. And thank you for everyone that supported this presentation. Do not hesitate contacting us using the emails on the screen. And we will now have a couple of minutes to answer your questions.